Welcome to the Deep Dive. We're here to basically pull out the key knowledge, the insights you need from recent research. Think of it as your curated download.
Yeah, exactly.
Today we're focusing on some really interesting papers in longevity biotech, stuff you might have touched on briefly in the Longevity Papers podcast a couple of weeks back.
Right. And the goal today isn't just to repeat those quick mentions. We really want to, dig into these studies properly.
Get under the hood a bit.
Exactly. Look at the findings, sure, but also how they got there, the methods, and importantly, bring a critical perspective. Things move so fast in this field.
Definitely. We need to understand what's really solid, what might need maybe a second look.
And the papers today cover quite a range. We're talking cellular aging, gut health links potentially, genetics, immune stuff.
Even broccoli.
Even broccoli, surprisingly. And circadian rhythms in muscle, which is where we'll start.
Okay, let's jump in. That first one really caught my eye, too.
Yeah.
It's called muscle peripheral circadian clock, drives nocturnal protein degradation via raised Rohr-River balance, and prevents premature sarcopenia.
Yeah.
By Kilu and Hughes in PNAS, May 5th, 2025.
This one offers a really fascinating angle on muscle aging. We often think of it as just wear and tear, right?
Yeah.
Passive decline. But this paper suggests there's actually an active regulation going on, controlled by the internal clock, right there in the muscle cells.
Right.
And their big finding, essentially, is that if you mess up this clock in zebrafish, you see premature muscle aging. Sarcopenia.
So it's not just about how much you use your muscles, but also their internal timing.
Precisely.
Right.
Which is kind of mind-bending, isn't it?
It really is. And the most counterintuitive bit, for me anyway, was this idea that actively breaking down protein at night, that's actually good for the muscle long term.
That's the core insight, yeah. It seems backwards at first glance. The paper really lays out how the muscle clock ramps up protein breakdown, specifically at night.
How do they show that? The model was zebrafish.
Zebrafish, yes. They engineered them with a disrupted muscle clock, and initially, these fish actually showed more muscle growth at night.
Okay, so far so good, Macy.
You'd think, but then fast forward to 24 months, which is getting old for a zebrafish, and those same fish showed clear signs of premature sarcopenia. Lower weight, shorter length, they were less active. It strongly suggests that initial nighttime growth boost, because the clock was broken, actually paved the way for earlier aging.
Wow. Okay, so how did they pin down the how? The cellular nuts and bolts.
They looked at the two main routes for protein breakdown. There's the ubiquitin proteasome system.
The cell's garbage disposal, basically.
Kind of, yeah. And then there's autophagy, where cells recycle their own bits.
Right.
And their data showed this nighttime peak in MRF, a key UPS player. Protein ubiquitination rates actually doubled at night.
Doubled? Okay.
And interestingly, if they inhibited MRF, guess what? Nighttime muscle growth increased.
Okay, so the UPS is definitely active at night, trimming things back. What about autophagy? Same story.
Pretty much. They saw ULK1, which kicks off autophagy, accumulating and becoming more active at night. Higher autophagic flux is the term.
Got it.
And just like with the UPS, if they blocked autophagy pharmacologically, nighttime muscle growth went up.
So it really looks like a coordinated effort driven by the clock to put the brakes on growth at night using both systems.
That's the picture they paint, a deliberate, timed process.
Okay, so the clock normally activates these breakdown pathways at night. How does breaking the clock lead to the premature aging they saw?
Right. This is where it connects to other aging hallmarks. In the fish with the wonky clocks, they found increased activity of TRC1.
TRC1, okay, that rings a bell. Big player in growth and metabolism.
Exactly. And it was specifically higher at night in these fish. Now, usually more TRC1 means more growth, but here, it seemed to put the brakes on autophagy.
Okay, so the high TRC1 was actually bad in this context.
It appears so. It inhibited ULK1. They saw fewer of these things called LC3 puncta, which you need for autophagy, and more buildup of P62.
Which signals that autophagy isn't working well, right? Stuff isn't getting cleared out.
Precisely. So the sequence seems to be: broken clock leads to high nighttime TORC1, which jams up autophagy, leading to a buildup of cellular junk. Classic aging sign. This failure in proteostasis, keeping proteins healthy, looks like a key driver.
And you mentioned earlier that the clock-disrupted fish didn't even respond to autophagy inhibitor.
Yeah, that's a telling detail. It really suggests the clock is upstream. If the clock's broken, that pathway's already messed up, so inhibiting it further doesn't do much.
Okay, that makes sense. It ties the clock directly to that proteostasis failure. What about figure 6? You said that pointed to something else important.
Yeah, figure 6 brings in another layer involving the balance between two nuclear receptors, roar and reverb. It seems NR1D1, which is reverb when overexpressed, actually promoted muscle growth. And even more directly, if they used a drug, SR90LM1, to activate reverb, it also boosted nighttime growth. It essentially mimicked the effect of breaking the clock.
So the natural daily rhythm, the back and forth between roar and reverb, is critical for keeping this protein turnover balance right.
That seems to be the implication, yes. Messing with that balance, pharmacologically or by breaking the clock, throws things off.
Okay, stepping back then.
Yeah.
What are the bigger picture implications here for aging, for longevity?
Several things, I think. First, this idea of chronotherapeutics for sarcopenia. Maybe timing exercise or even drugs could make a difference.
Aligning treatments with the body clock.
Right. Second, it really reframes protein breakdown. It's not just bad. Timed breakdown seems protective for muscle. Third, it reinforces that deep link between circadian rhythms and metabolism via TORC1 here. Mess one up, you affect the other.
And fourth.
The finding about early intervention being key. Fixing the clock early in the zebrafish larvae had the biggest impact. Suggests maintaining healthy rhythms early in life might pay dividends later for muscle.
That's quite profound. And the experiments sound pretty solid too. Zebrafish, the free-run conditions for true circadian analysis.
The OPP labeling to actually see protein turnover is neat. And yeah, following them for 2 years gives it weight.
What? Gotta wear the skeptic's hat. What are the potential caveats or limitations?
Well, number one, it's zebrafish. Great model. But human muscle might tick differently, the timing could vary.
True.
Also, they showed what happens when you break the clock, but they didn't test ways to fix it or restore that proper temporal balance. That's a missing piece.
Right. No intervention to rescue the phenotype.
And while they focused on roar reverb, other clock parts like cry and purr are surely involved too. And finally, the reduced activity in the old fish. Maybe some of that comes from the nervous system, not just the muscle itself that wasn't fully disentangled.
Okay, so if you were planning the follow-up studies, what's top of the list?
Oh, definitely human muscle biopsies. Take them around the clock, see if we see similar patterns in protein breakdown, clock genes. That'd be huge.
See if it translates.
Exactly. And then testing interventions in mammals. Can timed exercise or time-restricted feeding actually prevent muscle loss in aging mice, for instance?
Practical applications.
And digging deeper into the other clock components. What happens if you tweak cry or purr in muscle? Plus, those tissue-specific rescue experiments. Can fixing the muscle clock alone improve activity? Or is there muscle-brain feedback that's critical? Lots to explore.
It really is fascinating how this one study weaves together so many threads: time, protein quality control, tissue clocks, metabolism.
It does. And thinking clinically, it's just the focus, doesn't it? Maybe it's not just about build more muscle. Maybe it's about optimizing the rhythm of muscle maintenance.
Through timed exercise, timed eating, maybe even drugs targeting reverb someday.
Potentially. It opens up this idea of the circadian age of our muscle. Is its clock running well? That might be as important as how old it is chronologically. We've obsessed over protein synthesis, but this really hammers home that time degradation is just as vital.
Absolutely crucial insight. Okay, fantastic dive into that one. Let's shift gears completely now. Seaweed.
Seaweed, yes. Specifically, fucoidins from brown algae. This is a pooper. Fucoidins are xenotherapeutics that enhance SERT6-dependent DNA repair by Zhang et al. It was a preprint on Bioxaf, April 28, 2025.
Okay, xenotherapeutics compounds targeting senescent zombie cells. What's the core idea here?
The headline finding is that these fucoidins seem to reduce cellular senescence, and they appear to do it by activating an enzyme, SRT6.
RT6, involved in DNA repair and epigenetics, right? Needs NAD plus hand.
That's the one. So the claim is that fucoidins activate SRT6, which then boosts DNA repair, and this helps clear out senescent cells, at least in accelerated aging mouse models. They're positioning fucoidins as potentially the first natural product xenotherapeutic working via SRT6.
Okay, that sounds pretty significant. A natural compound hitting senescence and DNA repair through a key pathway like SRE6.
That's the potential excitement, yes. SRT6 is linked to reducing the inflammatory SSP from senescent cells and also maintaining genomic stability. So you could theoretically tackle inflammaging and DNA damage at once.
A double whammy.
Potentially. And the paper suggests they're orally bioavailable in mice and seemed safe, which always helps translational prospects.
Right. But skepticism time. What made you pause or raise an eyebrow?
Well, one thing is the mechanism. They seem to link it partly to autophagy via something called NCOA4, but also to SRT6. The exact interplay and whether SOT6 is truly the main driver wasn't crystal clear. It might need more work to be sure it's not just, correlation.
Okay, mechanism needs tightening. What else?
A big one is the model. They showed lifespan benefits, but only in accelerated aging mice, prone to aging faster.
Not normal aging.
Right. We don't know if fucoidins actually extend lifespan or health span in normally aging animals. That's a critical missing piece for relevance to, well, normal aging.
Huge difference, yeah.
And finally, fucoidins themselves. They're complex molecules. Depending on the seaweed source, how you extract them, their structure, especially the sulfation pattern, can vary a lot.
Meaning one batch might work differently from another.
Exactly. That variability is a real challenge for natural products. You need standardization to know if the results are broadly applicable.
Okay, valid points.
Yeah.
So follow-up experiments. What's needed?
Top of my list. Test it in normal wild-type aging mice. Proper dose-response studies. Do they live longer? Healthier? That's essential.
The gold standard validation.
But then nail down that SRT6 dependence. Use SRT6 knockout mice. If the fucoidins don't work in mice lacking SRT6, that strongly supports the mechanism.
Okay.
And finally, given the SRT6 NAD plus connection, it would be really interesting to compare fucoidins directly to known NAD plus boosters like NR or NMN. How do they stack up in terms of effects and mechanisms?
Good comparison.
Yeah.
I see the source material also mentioned an AI analysis gave this paper a high relevance score.
It did, yeah. Apparently because it hits multiple aging hallmarks and showed some multi-tissue benefits. Which is interesting. AI can definitely spot patterns.
Right.
But it's based on the data in the preprint. It doesn't replace the need for those rigorous follow-up experiments we just talked about. It flags potential but doesn't confirm it.
Got it. Okay, so 2 really meaty papers unpacked. But you mentioned there was a flurry of other relevant stuff recently, too.
Absolutely. The field is just buzzing. We can't go deep on all of them, but definitely worth mentioning a few highlights.
Give us the quick rundown.
Okay, there was a massive single-cell paper from Lewinen and colleagues on Biorxiv, an epigenomic atlas looking at chromatin accessibility.
How massive are we talking?
Around 7 million cells across 21 mouse tissues. Just huge. It gives this unprecedented map of how the epigenome changes with age systemically, but also in very specific cell types. Could be a goldmine for finding new aging biomarkers.
Oh, that sounds like a foundational resource for the community. What else?
There was a big genetic study from Mullis et al., also Biorxiv, used diversity outbred mice, genetically very varied. They mapped genes linked to lots of age-related conditions.
Funny surprises.
One interesting thing was a surprisingly weak link between the genes influencing overall lifespan and the genes influencing frailty.
Oh, so living long doesn't automatically mean avoiding frailty, genetically speaking.
Seems that way. Or at least the genetic drivers might be somewhat distinct. Makes you think about how we measure successful aging.
Definitely. And you mentioned broccoli earlier.
Ah, yes. The sulforaphane paper, Sidor et al., by Orksorp again, found that sulforaphane extended lifespan in C. elegans worms.
Worms, okay. Important caveat.
Big caveat. But interestingly, they linked it to slowing down a transcriptional aging clock, measuring age based on gene activity.
Okay.
And like the muscle clock paper, they found early life treatment was most effective.
Interesting parallel. Anything else stand out?
2 more quick ones. Kamat et al. In Science Advances used AI to classify senescent cells based purely on their shape, their morphology.
Just by looking at them.
Essentially, yes, with sophisticated imaging and AI. And they found different shape-based subtypes responded differently to senolytic drugs, which hints at
More personalized senolytic therapy down the line, targeting specific types of senescent cells.
Exactly. Could make treatments more effective or reduce side effects. And finally, another AI paper of Chaishiev et al. In Aging Cell. They used AI to find combinations of existing drugs.
Cocktails.
Yeah, polypharmacology. Drug cocktails that significantly extended lifespan in worms by hitting multiple aging pathways at once. Showcases AI's power in drug discovery for aging.
That's quite a landscape, from
Right.
single cells to AI drug design. But important to keep the skeptical notes in mind too, right?
Absolutely. As the source material pointed out, a lot of work is incremental, focusing on one pathway. Worm results need mammal validation, and some senolytic work might be finding compounds that work similarly to ones we already know.
So, progress, but keep the critical thinking engaged.
Always. It's exciting, but requires careful validation every step of the way, especially before thinking about humans.
Okay, let's try and wrap this up. What are the main headlines from this deep dive for you?
I think that muscle clock paper really stands out. The whole idea of timed protein breakdown being protective is, well, it's a paradigm shift, potentially. Really novel.
Yeah, that temporal regulation aspect is key.
And the fucoidin paper, despite the caveats, points towards a potentially interesting new class of natural xenotherapeutics acting via SRT6. Needs more work, but the concept is intriguing.
And just the sheer breadth of approaches we touched on, epigenomics, genetics, AI, cell morphology, it really underscores that aging is complex. No single magic bullet.
Definitely not. It's about understanding this incredibly intricate network. And the key message for you, listening, is to follow this exciting research, but always with that critical lens. Celebrate the breakthroughs, but understand the need for rigorous follow-up.
Right. Especially before jumping to conclusions about human health.
Couldn't agree more.
So maybe a final thought to leave everyone with. Considering that muscle clock paper, that intricate timing, could focusing on optimizing our daily rhythms, sleep, food timing, exercise timing, be a much more powerful lever for longevity than we've really appreciated?
Oh, it's a compelling thought, isn't it? Basic lifestyle, but timed precisely.
Or are we on the cusp, with AI accelerating discovery like in that worm study, of finding really potent multi-target drug cocktails that could dramatically reshape aging?
Two very different but equally fascinating possibilities for the future. Definitely something to ponder as the research continues to unfold.
A lot to think about. Thanks for joining us for this deep dive.